Very good morning students. So this is your Prasanna ma'am from NEET World, and today we are going to discuss about the biological classification. As we have discussed in our previous class that I am beginning the core botany concepts of which we have dealt with living world. Today we are going to discuss on the biological classification, of which today we are going to cover kingdom systems of classification and kingdom Monera. Before entering into the concept, let me first tell you like what sort of questionnaire can we expect from this chapter and what are different types of or different styles of the questions that arise from this chapter. Like we may expect match the following question, mostly example based questions beta, mostly example based questions, example based, statement based, assertion reason. So these type of questions we may expect from this chapter, and the range of questions can be 2 to 3 number of questions can arise from this chapter biological classification, okay, of which today whatever we are dealing this kingdom systems of classification, we may expect match the following type of questions also, giving us the scientist name and the system of classification, and about kingdom Monera also totally we are going to discuss. And before telling you about kingdom Monera, we have to learn kingdom Monera along with bacteria physiology, or from the cell biology chapter we have to learn it along with prokaryotic cell, because the questions from Monera can be asked in connection with the prokaryotic cell of cell biology, okay. So today we will discuss about kingdom systems of classification and kingdom Monera. Kingdom Monera you have to revise along with the prokaryotic cell of cell, the unit of life. So this is clear to you, right beta? Let's get started. So here when you have to look into the kingdom systems of classification, we have the first scientific method of classification is given by Aristotle. First we have to look into the Aristotle's classification, and then 2 kingdom systems of classification, 3 kingdom, 4 kingdom, 5 kingdom and 6 kingdom. Of all these kingdom systems of classification, the most widely accepted system of classification is Whittaker's 5 kingdom system of classification. So first let us look into the Aristotle classification. As I told you, Aristotle has gave us the first scientific method of classification, wherein he has classified the living organisms into plants and animals. Plants based on their identity, based on their morphological features, just he has classified the plants into 3 types. Those are herbs, shrubs and trees. This is just based on simple morphological characters, whereas animals are classified into those which has RBC and those which do not have RBC. Those animal cells which do not have RBC, they are anemic. Those living cells which has RBC, those are anemic, okay, anemia and anemia, do remember. So yes, the first scientific method of classification is given by Aristotle. He has classified the plants based on simple morphological characters into herbs, shrubs and trees, and animals based on the presence and absence of RBC into anemia and anemia, that is with RBC and without RBC. So this is about simple Aristotle's classification beta. So if there is a question like who gave us the first scientific method of classification, then your answer should be Aristotle's system of classification. Then comes 2 kingdom system of classification, which is given by Carolus Linnaeus. So it is the scientist Linnaeus who has proposed 2 kingdom system of classification. So in this 2 kingdom system of classification, many criteria have been considered. Very good morning ma'am. Hi. So go through the class. Any doubts anywhere, you can drop a message in the chat box ma'am. So first let us see the criteria. What are the criteria of Linnaeus 2 kingdom system of classification? We have 5 criteria, okay. First is cell wall, mode of nutrition, Third, response to external stimuli, locomotion, contractile system, and then examples. So, these are the 5 criteria. Okay. Cell wall, mode of nutrition, response to external stimuli, locomotion and contractile system. Of these 5 criteria, which is the most important criteria? The most important criteria is cell wall. Okay. Cell wall is the major criteria for two kingdom system of classification. Okay. Cell wall is the major criteria for two kingdom system of classification. In Plantae, cell wall is present. Those cells, be it prokaryotic, eukaryotic, unicellular, multicellular, all those organisms which have cell wall are placed under the kingdom Plantae. Okay. And those that doesn't have cell wall are placed under the kingdom Animalia. Next is what ma? Mode of nutrition. Those living organisms in which the mode of nutrition is autotrophic are placed under Plantae, heterotrophic under Animalia. Response to external stimuli, it is slow in plants and fast in Animalia kingdom. Locomotion is absent in Plantae, present in animal kingdom. Contractile system is absent in Plantae, present in Animalia. Examples, as I told you, be it prokaryotic or eukaryotic, unicellular and multicellular, autotrophic or heterotrophic, all those organisms which have cell wall are placed under Plantae. That is the reason I told you what is the major criteria for two kingdom system of classification, that is cell wall. So, the examples under Plantae include bacteria, algae, fungi and plants. For Animalia, protozoa, vertebrates and invertebrates. This is in brief about two kingdom system of classification. The question we may expect is what is the major criteria for two kingdom system of classification, that is cell wall. Remember that beta. And most importantly, what are the major drawbacks or objections of two kingdom system of classification, that is important. What are the drawbacks of two kingdom system of classification? This in our NCERT, the lines clearly state the differences between the drawbacks of two kingdom system classification and comparing with the 5 kingdom system of classification, it is clearly dealt. So, first let us look into the drawbacks of two kingdom system of classification. Prokaryotic bacteria, that is blue green algae, is placed along with the eukaryotes, that is algae, bryophytes, pteridogymna and everything. Okay. So, prokaryotes and eukaryotes are placed under Plantae kingdom only, that is one drawback. Second is unicellular organisms like Chlamydomonas are placed along with the multicellular algae like Spirogyra, which is filamentous. Okay. So, unicellular and filamentous algae are placed under one kingdom again. Third is photosynthetic green algae are placed along with non-photosynthetic fungi, because fungi also has cell wall which is made up of chitin. So, these are the major drawbacks of two kingdom system of classification, highly important. We can expect a question directly from the NCERT liner. Okay. So, these you have to remember beta. Next, three kingdom system of classification. Three kingdom system of classification is proposed by Ernst Haeckel. Okay. Ernst Haeckel has proposed three kingdom system of classification, and the new kingdom he has introduced is Protista. Along with Plantae and Animalia, he has introduced Protista. Protista is the new kingdom he has introduced, and he has placed all the unicellular eukaryotic organisms. Firstly, unicellular organisms only by Ernst Haeckel, all the unicellular organisms are placed under the Protista. Fine. So, here we can expect two questions. Three kingdom system of classification is proposed by Ernst Haeckel, and what is the new kingdom he has introduced? Protista. What type of organism he has placed in the Protista? Unicellular organisms he has placed under the kingdom Protista. Till here it is fine, Navita. Next, co-plant. Co-plant has given us the 4 kingdom system of classification, and the new kingdom he has introduced is Monera. Previously, Ernst Haeckel has placed all the unicellular organisms under Protista, but here all the unicellular prokaryotic organisms, or you can simply say prokaryotic organisms, are placed under Monera here. Okay. All prokaryotic organisms are placed under the kingdom Monera, and co-plant introduced the new kingdom, that is Monera, in 4 kingdom system of classification. So, 4 kingdom system of classification is given by Copeland. The 4 kingdoms he has given is Monera, Protista, Monera, Protista, Plantae and Animalia. In Monera is the new kingdom he has introduced, and he has placed only prokaryotic organisms in the kingdom Monera. Next, the widely accepted model, that is R.H. Whittaker's 5 kingdom system of classification. So, in this 5 kingdom system of classification, what are the major criteria that he has considered? The major criteria considered over here is cell structure, body organization, body or thallus organization, mode of nutrition, reproduction and phylogenetic relationships. So, here we have cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. These are the major 5 criteria of 5 kingdom system of classification. Of these 5 criteria, what is the most important criteria considered for 5 kingdom system of classification? That is, mode of nutrition is the most important criteria considered for 5 kingdom system of classification. And here if you see with Monera includes all unicellular prokaryotes, Protista eukaryotic unicellular organisms are introduced. Next, fungi multicellular eukaryotes, Animalia multicellular eukaryotic, Plantae also multicellular eukaryotic species are included. So, this is about criteria of 5 kingdom system of classification. Let us look into what is the kingdom introduced in 5 kingdom system of classification. The kingdom which is introduced in 5 kingdom system of classification is fungi. Okay. Fungi is introduced in 5 kingdom system of classification. Let us quickly recall 2 kingdom system of classification introduced by Linnaeus, and what are those 2 kingdoms beta? Plantae and Animalia. 3 kingdom is given by Ernst Haeckel, okay, and the new kingdom he has introduced is Protista, wherein he has placed all the unicellular organisms. Third, 3 kingdom system of classification I have dealt, it is by Ernst Haeckel. Next, 4 kingdom system of classification by Copeland. The new kingdom he has introduced is Monera. Okay, Monera he has introduced the new kingdom, wherein he has placed all the prokaryotic organisms. And 5 kingdom system of classification, new kingdom introduced is fungi. Till here it is clear, right now? Now, amongst this is the NCERT table, which is very important. So, from 5 criteria that we have discussed for Whittaker's 5 kingdom system of classification. First is what, Nana? Cell structure. So, cell structure we have cell type and cell wall. These two are talking about cell structure. Next, cell structure, this also includes nuclear membrane. So, cell type, cell wall, nuclear membrane comes under what criteria? Cell structure. Next is what? Body organization or thallus organization. And then third, that is the most important criteria of 5 kingdom system of classification, that is mode of nutrition. So, here if you see cell type in Monera, it is what? Prokaryotic cell type. In Protista, eukaryotic cell. Fungi is also eukaryotic. Plantae eukaryotic. Animalia eukaryotic. When it comes to cell wall, Monera has non-cellulosic cell wall. Mostly bacteria are the sole members of the kingdom Monera, and the cell wall of eubacteria will be peptidoglycan, which is made up of polysaccharides and amino acids. Okay, polysaccharides and amino acids. Next, so cell wall is present in Protista also, those protistans which are plant like. Next, in fungi also cell wall is present, which is chitin, commonly called as what fungal cellulose. Okay, so this is commonly called as fungal cellulose. Plantae also cell wall is present, which is made up of cellulose, and in Animalia cell wall is absent. Remember the cell wall and its composition, very important. Next, nuclear membrane is absent in Monera. It is present, nuclear membrane is present in all the 4 kingdoms because they are eukaryotic. Next comes the body organization. Cellular type of body organization is seen both in Monera and Protista. Cellular body organization which are prokaryotic, and this is eukaryotic cellular organization. And in fungi, this is multicellular and it has loose tissue. Plantae, tissue and organ type. Animalia, tissue, organ and organ system type. Next, mode of nutrition. Autotrophic mode of nutrition, which includes both chemosynthetic and photosynthetic. Heterotrophic nutrition includes saprophytic, parasitic, okay, mode of nutrition in Monera. So, of these 5 kingdoms, which will show you the more nutritional diversity or metabolic diversity, then comes the answer about Monera. Monera kingdom will show high metabolic diversity, be it nutrition or respiration. So, high nutrition diversity is shown by which kingdom? Monera. So, yes ma'am, in 2025 question was arised from this chart. Yes ma'am, question will definitely arise from this chart, but you have to look into this chart in a different perspective. Like how I have told you, amongst 5 criteria, only 3 are given, like cell type, cell wall and nuclear membrane comes under what? Cell structure only, and then body organization, mode of nutrition. Clear, right? So, this chart tells you about more information. Okay, you have to connect the theory which is given in the NCERT above the chart and below the chart and look into the chart. Okay, so next, Protista, it also includes 2 types of nutrition, autotrophic and heterotrophic. Fungi only heterotrophic nutrition, Plantae autotrophic, Animalia has only heterotrophic nutrition, which has holozoic type of nutrition, whereas Plantae holophytic type of nutrition. So, this is all about 5 kingdom system of classification. Next, Monera, if you see, all prokaryotic organisms are placed under Monera. Protista, which is the common ancestor for fungi, Plantae and Animalia. Protista is the common ancestor for fungi, Plantae and Animalia. Remember that ma. Next, 5 kingdom system of classification is brought about by R.H. Whittaker. In 5 kingdom system of classification, if you have to see, under Protista, Chlamydomonas and Chlorella are placed along with Paramecium and Amoeba. This is your NCERT liner, very much important. Okay. So, Protista is standing as a common ancestor for Plantae, Animalia and fungi. So, in 5 kingdom system of classification, Chlamydomonas and Chlorella, which are earlier placed in algae, are brought together with Paramecium and Amoeba. This does not have cell wall, whereas Chlamydomonas and Chlorella has cell wall. So, these 2 are placed in what? Protista. That is why we have 3 types of protists, right ma? Plant like protists, fungi like protists and animal like protists. Fine. So, remember this. Okay. Next, fungi. Fungi is the new kingdom which is introduced. It has cell wall, it is multicellular and heterotrophic. Fine. Fungi has cell wall, it is multicellular and it is heterotrophic. Next, Plantae. Plantae has cell wall and the nutrition is autotrophic mode of nutrition. Animalia does not have cell wall and it is having heterotrophic mode of nutrition. These are the keynote points that you have to remember with respect to each and every kingdom of classification. Till here it is fine, right Vita? Next, 6 kingdom system of classification. 6 kingdom system of classification is given by Carl Woese. 6 kingdom system of classification is also called as 3 domain system of classification, wherein he described that there is a common ancestor called as progenote. Okay, there is a common ancestor called as progenote. It has 16S rRNA. Okay, there is a common ancestor, that is progenote, which has 16S rRNA in common, and 3 domains from this common ancestor, those are Archaea, Bacteria and Eukarya. Okay, it has Archaea, Bacteria and Eukarya. Archaea, if you see, the cell wall of Archaea is made up of polysaccharides and proteins. Okay, cell wall of Archaea has what? Polysaccharides and proteins. Cell membrane has branched chain lipids. And here under Archaea domain we have only one kingdom, that is Archaebacteria. Second is what? Bacteria. This domain cell wall is made up of what? Peptidoglycan. So, cell wall is distinguishing between the prokaryotic organisms into Archaea domain and Bacteria domain. Remember that. And under this Bacteria domain you have only one kingdom, that is Eubacteria. Next domain is Eukarya. Here you will be having 4 kingdoms. Those are Protista, Fungi, Plantae and Animalia. Okay, Protista, Fungi, Plantae and Animalia under eukarya. So, till here it is clear, right now. So, under 6 kingdom system of classification, which is also called as 3 domain system of classification, which is given by Carl Woese, he says that there is a common ancestor called as progenode, which has 16 S rRNA. It has 3 domains: archaea, bacteria and eukarya. Archaea, cell wall is made up of polysaccharides and proteins. Cell membrane has branched chain lipids. It has 1 kingdom, that is Archaebacteria. Bacteria domain has cell wall peptidoglycan. It has only 1 kingdom, that is Eubacteria. Next, Protista, eukarya domain, it has 4 kingdoms: Protista, Fungi, Plantae and Animalia. So, this is all about 6 kingdom system of classification. Now, as we are discussing about 5 kingdom system of classification, that is Whittaker's 5 kingdom system of classification is widely accepted, right. So, in this chapter we are going to discuss or exercise each and every kingdom in detail, okay. That is from Monera, Protista, Fungi, Plantae and Animalia. Of course, Plantae and Animalia we have 2 separate chapters, plant kingdom and animal kingdom. These 3 we are going to look at here now, and in this chapter at the end we are also going to discuss about those organisms which are not included in Whittaker's 5 kingdom system of classification. Those like virus, viroids, prions, lichens, which are not included in the 5 kingdom system of classification. About this also we have to discuss at the end of this chapter. Clear right now? So, now let us begin with the kingdom Monera. First, prokaryotic, they does not have a nuclear membrane, no nuclear membrane, nucleolus and chromatin. We know this, right. Next, histone proteins and membrane bound organelles are also absent. So, to hold on the chromatin inside the cell, histone proteins are not present, some positively charged proteins are present, okay. Rather than histone proteins, some positively charged proteins are present inside the cytoplasm of the bacteria to hold on the chromatin, which is negatively charged. So, what are absent? Note down this keenly, that nuclear membrane, nucleolus, chromatin, histone proteins, membrane bound organelles are absent. Next, rigid cell wall is present. There is rigid cell wall for different classes of bacteria. For example, Eubacteria is having what cell wall composition? Peptidoglycan. Archaebacteria has polysaccharides and sugars. Mycoplasma, there is no cell wall. This is wall-less bacteria. Next, actinomycetes, the cell wall is made up of mycolic acid. So, do remember the cell wall composition of each and every prokaryotic bacterial type is very important, okay. So, Eubacteria which has peptidoglycan, Archaebacteria polysaccharides, Mycoplasma is wall-less, it does not have any cell wall, Actinomycetes has the cell wall composition is mycolic acid. Next question, next you look into nucleoid, that inside the cytoplasm there is no membrane bound nucleus, but then chromatin is not completely dispersed into the cytoplasm, it is present at one area in the cytoplasm, that is called as nucleoid, which is also called as incipient nucleus, nucleoid or incipient nucleus, which is composed of double stranded circular DNA, RNA and proteins, okay. So, it has double stranded circular DNA, RNA and polyamines. As I told you, there are no histone proteins over here, it has polyamines. So, each and every point that I am discussing is highly important for your exam beta, note it down keenly, okay. Next, this you see, as I told you, we have to learn the kingdom Monera in connection with prokaryotic cell of your cell biology, right. So, here we have double stranded circular DNA which is with the polyamines. So, this is nucleoid or incipient nucleus. Along with this, we have one more extra chromosomal DNA, that is plasmid DNA, right ma. We have dealt with plasmid DNA also, which is highly crucial to describe the phenotypic characteristic of the bacteria, and it will also provide the antibiotic resistance to the bacteria. And what are the non-membrane bound organelles? Those are ribosomes. So, here we have what ribosomes? 70S ribosomes, 2 types of ribosomes we have. One is cytosolic ribosome, second is the ribosomes which are attached to the cell membrane, okay. Cell membrane, the ribosomes are attached. This is for what? Protein synthesis. We know the ultimate function of ribosome is what? Protein synthesis. Cytosolic ribosomes will synthesize the proteins, okay, whereas those ribosomes which are attached to the cell membrane, they will synthesize the proteins which are helpful in translocation. Remember that. Next, so here ribosomal type is 70S, we know larger subunit will be 50S, smaller subunit will be 30S. So it exhibits high metabolic diversity. High metabolic diversity will be exhibited. Nutrition, if you take, we have autotrophic nutrition and heterotrophic nutrition. As I already told you, the highest nutritional diversity is exhibited by what ma? Monera kingdom only. So we have two types of nutritions, those are photosynthetic and chemosynthetic. Heterotrophic nutrition, we have saprophytic, symbiotic and parasitic modes of nutrition. Look at it keenly with the respiration. Also we have two types, aerobic and anaerobic. Aerobic again obligate aerobes, those will oxidize the food completely in the presence of oxygen only. So oxygen is mandatory, if oxygen is not present they cannot oxidize the food material. So those are obligate aerobes. Facultative anaerobes means what ma? Like facultative, look it with the synonym of sometimes. Sometimes they are acting as anaerobes, okay. So they can respire even in the aerobic conditions. When aerobic conditions are not present, they can even respire in the anaerobic condition. Next, anaerobic mode of nutrition. Here we have obligate anaerobes again, facultative anaerobes also. Reproduction, if you take ma, asexual, sexual mode of reproduction. Sexual reproduction we will not call it as, because there is only one genome involved in this. So, right, we will call it as sexual recombination or gene recombination. As such we have three ma, conjugation, transformation and transduction. So, look at here, bacterial appendages. We have two types of bacterial appendages, one is motile and second is non-motile. Motile, it is flagella, non-motile we have pili and fimbriae, right. So, this is all about the general characteristic features of the bacteria. Now, let us look into the classification of the Monera. Based on the presence or absence of cell wall, habitat, type of the cell wall, okay, presence or absence of the cell wall, habitat, type of the cell wall, we have four different types of the Monera. Those are Archaebacteria, Eubacteria, Mycoplasma and Actinomycetes. So, Archaebacteria you see ma, these are the most primitive monerans and they occur in the extreme or harsh habitats, okay. They are the most primitive monerans and they occur in extreme or harsh habitats. So, based on the habitat we have halophiles, thermoacidophiles and methanogens based on the habitat, okay. So, here halophiles if you see, those Archaebacteria, they grow in extreme salty areas, okay, they grow in extreme salty areas. Thermoacidophils, okay, they survive, the habitat is in hot spring and they tolerate high temperature and high acidity, okay, they tolerate high temperature and high acidity and low pH. That is about thermoacidophils. Methanogens, they grow in marshy areas and they are present in the gut of ruminants. Remember that. So, this is based on the habitats, they are classified based on the habitat. Now, halophiles, okay, halophiles best examples include Halobacterium and Halococcus. Thermoacidophils best examples, Thermoplasma and Sulfolobus. Methanogens examples, Methanococcus and Methanobacteria. I am again repeatedly telling you examples are highly important, we cannot skip even a single example over here. So, they grow in what? Marshy areas. They live in the gut of ruminants like cow, buffalo which ruminate all the time. So, methanogens will convert the carbon dioxide, methanol and formic acid into methane, which is the production of biogas from dung of animals, okay. So, this is all about the types of Archaebacteria. Now, when we have to discuss about Eubacteria, we all know bacteria are the sole members of the kingdom Monera and Eubacteria, these are cosmopolitan in distribution, they are present everywhere. So, here if you look into the cell wall beta, it is composed of murin, okay, cell wall is composed of murin, peptidoglycan, and it has mesosomes. Mesosomes we have discussed, right, in cell biology. Mesosomes of a prokaryotic cell are equivalent to what organelle of eukaryotic cell? Mitochondria, because mesosomes are respiratory in function. Next, ribosomes, which are 70s, they are keenly involved in what? Protein synthesis. And motile structure, only one type of motile structure in prokaryotic cell, that is flagella, whose microtubular arrangement will be 9+0 microtubular arrangement. Here you see based on the shape, bacteria is of many types. It is grouped into 4 categories: cocci, bacilli, spirilla and vibrio. Cocci are spherical shaped, bacilli rod shaped, spirilla spiral shaped, vibrio comma shaped. So, bacilli are most common form of bacteria over here. Okay, these are rod shaped. Metabolic diversity, as I told you, anabolism includes nutrition, catabolism includes respiration. So, nutrition, as I have told you, it is autotrophic and heterotrophic mode of nutrition. Autotrophic we have photoautotrophic and chemoautotrophic. Heterotrophic, symbiotic, parasitic and saprophytic. Look at here photoautotrophic and chemoautotrophic. One thing you remember, photo means what? Sunlight. So, here the energy used is purely sunlight for the synthesis of food. But here chemoautotrophic, energy is derived from some chemical substance, and here they rely completely on ATP. For chemoautotrophic nutrition, the energy utilized is ATP. For photoautotrophic nutrition, energy used is sunlight. And here the electron donor for photoautotrophic nutrition is water if it is conducting oxygenic photosynthesis. If it is conducting anoxygenic photosynthesis, then the electron donor will be H2S. Here you see here it is the inorganic electron donor will be present like hydrogen ions, sulfate ions, ferrous ions, nitrates, nitrites. These are the inorganic electron donors. As it is utilizing the inorganic electron donors, they play crucial role in recycling the nutrients. Okay, they play crucial role in recycling nutrients like nitrogen, phosphorus, ferrous and sulfur. Okay, here the electron donor is water if it is conducting oxygenic photosynthesis, H2S if it is conducting anoxygenic photosynthesis. Examples over here for photoautotrophic nutrition include chloronema and chlorobium. Chloronema and chlorobium are included under photoautotrophic nutrition. Nitrifying bacteria, they are the best example for what? Chemoautotrophic nutrition. Clear right now? So, here first if you have to see ammonia first they get converted to nitrates, okay, by the bacteria. Those are Nitrosomonas and Nitrosococcus. So, they are relying on nitrates. These are the best examples for what? Chemoautotrophic nutrition. Next, nitrate turns to what? Nitrites by what? Nitrobacter and Nitrocystis. So, this is about the photoautotrophic and chemoautotrophic. Do remember the examples. Chloronema and chlorobium over here, and here it is nitrifying bacteria, and energy used for both of them, electron donors involved for both of them. This is very important. Next, heterotrophic mode of nutrition if you see saprophytic, symbiotic and parasitic. Saprophytic mode of nutrition, majority of them are heterotrophs and they secrete digestive enzymes. Okay, they secrete digestive enzymes where they will convert the complex insoluble substances to simple soluble compounds. Okay, complex insoluble substances will be converted to simple soluble compounds. Symbiotic mode of nutrition, if that is there, then there will be mutual benefit. Okay, so Rhizobium leguminosarum and leguminous plants, they show symbiotic association, wherein leguminous plants will provide shelter to the Rhizobium leguminosum and Rhizobium will help will act as nitrifying bacteria, okay, for leguminous plants. Next, parasitic mode of nutrition, best examples include E. coli and Salmonella. Here what is the most important point to note down is examples, be it autotrophic or heterotrophic. In autotrophic, photoautotrophic, chemoautotrophic, examples of that. And here saprophytic, symbiotic, parasitic and examples of that are highly important. Next, respiration. aerobic and anaerobic mode of respiration. Here if you see obligate aerobes, as I told you, they oxidize the food completely in the presence of oxygen. So, here 100% of oxygen is required. Best example is Bacillus subtilis. Okay. And here if you see facultative anaerobes, means most of the time they are aerobes, sometimes they are acting like anaerobes. Best example is what? Pseudomonas. Next, anaerobic bacteria if you have to consider, obligate aerobes and facultative aerobes. Obligate aerobes, Clostridium. Facultative aerobes means most of the time they are anaerobic, sometimes they act as aerobes. Best example will be Chlorobium. Okay. So, most of the photosynthetic bacteria are facultative aerobes only. Okay. Most of the photosynthetic bacteria are facultative aerobes only. Okay. Next, now when we have to talk about eubacteria, best example given in our NCERT, that is blue green algae, that is Cyanobacteria. First look at its structure carefully. So, here if you see, it is a filamentous blue green algae, wherein it can be unicellular, colonial or filamentous, any of the forms it can be Cyanobacteria. But what type of cells we have? We have two types of cells over here. Those are vegetative cells and heterocysts. We have vegetative cells and heterocysts, and these are covered or encapsulated by what? Gelatinous sheet. Okay. These are uncovered by what? Gelatinous sheet, and it has vegetative cells and heterocysts. Vegetative cells will be having two photosystems, photosystem 1 and photosystem 2, and they have chlorophyll A. Electron donor will be water. So, they are conducting what photosynthesis? Oxygenic photosynthesis in the vegetative cell. This, from here, this concept we can expect a connecting question between this BGA and the photosynthesis. Okay. So, what type of photosystems does it have in vegetative cells? PS1 and PS2, chlorophyll A is present, electron donor is water, and it conducts oxygenic photosynthesis. And they have some colored pigments like mesosomal extensions in blue green algae are called as chromatophores. This we have learnt in cell biology. Okay. Mesosomal extensions with colored pigments in BGA are called as chromatophores. Next is heterocyst. Heterocyst will have only photosystem 1. This will generate ATP, and this is used for nitrogen fixation. The walls of the heterocyst are impervious for gaseous exchange. Okay. And this is oxygen intolerant. Okay. Heterocyst is oxygen intolerant, whereas vegetative cells will conduct oxygenic photosynthesis. How is it? Because heterocysts are purely involved in what function? Nitrogen fixation. And for nitrogen fixation, we need the enzyme nitrogenase. Okay, the enzyme we need for nitrogen fixation is nitrogenase. Nitrogenase will function only in the anaerobic conditions. Remember that. And here we have colored chromoplasm and colored peripheral chromoplasm, and at the center non-colored centroplasm is present. This is about the cytoplasm of the vegetative cells. And we also have some broad cells in between, okay, which are called as akinates. They are purely helpful for the reproduction, vegetative reproduction of BGA. Okay. So, this is all about the structure of blue-green algae. It has two types of cells, vegetative and heterocyst. Vegetative cells purely involved in oxygenic photosynthesis. So, that is why they have this pigment, photosystems, electron donor. Remember that. And next, chromatophores, mesosomal extensions in BGA with pigments are called as chromatophores. And then heterocyst, whose membrane is impermeable for oxygen because it has function, that is nitrogen fixation, where the enzyme is nitrogenase. And then akinates, which are dead cells, which are purely helpful for what? Vegetative reproduction or vegetative propagation. And here you see we have unicellular, colonial, filamentous, aquatic or terrestrial algae, photosynthetic autotrophs. Mostly they are primitive organisms to conduct oxygenic photosynthesis. Few of the BGA members, they form algal blooms. Okay, they form algal blooms in polluted water bodies because of what? Eutrophication. Because of eutrophication, they form algal blooms in polluted water bodies. Heterocysts, these are the specialized cells to fix atmospheric nitrogen, for example, nostoc and anabina. Here about achinites, I told you, right, achinites are thick walled dormant cells in blue green algae. They germinate new fragment. Trichodesmium erythraeum is one of the marine cyanobacteria. This will make the red color, this will create the red color of the Red Sea because of its pigmentation, because of the toxins that it is releasing. Okay. So, Trichodesmium erythraeum, this is a marine cyanobacteria which gives the red color of the Red Sea, and we have one more unicellular eukaryotic from Protista, that is Goniolax, which is under dinoflagellate, we will learn. Goniolax also will cause red tides of Mediterranean Sea. Distinguish between these two examples beta. Trichodesmium erythraeum is a BGA, Goniolax is a dinoflagellate, that is Protista. Lastly, about reproduction, we have dealt or we have discussed that two types of reproduction will be there, asexual reproduction and sexual reproduction. Asexual, under favorable conditions, it will conduct what? Binary fission only, which is most common. Unfavorable conditions, it has what? Endospores. Okay. And sexual recombination, we have conjugation, transformation and transduction. Binary fission, if you see, which is the most common, binary fission is also called as amitosis because there is no spindle fiber formation over here. And endospores, if you see ma, endospores have thick cortex layer which is made up of calcium dipicoic acid. Endospore will serve as organs of perennation but not reproduction. This has low water content, low metabolic activity, it is unharmed. This is unharmed during pasteurization. Endospores are means of perennation but not reproduction. Remember, all these points are important. Next. So, in favorable conditions, what does endosperm do? That will release out bacteria. From one endospore, one bacteria only will come. Sexual recombination, there is no gamete formation or gamete fusion over here. So, there is no gamete formation or gamete fusion, that is why we are not calling it as sexual reproduction, rather we are calling it as sexual recombination. We have three types as such: transformation, transduction and conjugation. Transformation, this is best explained through Griffith's experiment on Streptococcus pneumonia, where two strains are taken, one is R strain and S strain. R strain is called as rough coat, it has rough coat, and S strain has smooth coat over it. So, here if you see, when S strain, that the Streptococcus bacteria which does not have any rough coat, it has smooth coat, when it is given to the mice, when smooth S strain is given to the mice, remember that S strain is virulent, R strain is non-virulent, because S strain, it has smooth shiny coat around the bacteria, it can survive in the gut line, it can survive in the host cells. Okay. Whereas rough strain, that is R strain, it does not have any protective encapsulation around the bacteria. So, the bacteria will be dying and the host will be remaining alive. As such, when we have given S strain to the host, mouse dies because it is taking the S, when S strain is given. When R strain is given to the living mice, it is survived. S strain heat killed, mouse is alive. When R strain is mixed with the S strain, okay, R strain is mixed with the heat killed S strain, R strain gets virulent and the mouse dies. So, here there is no contact between the donor and recipient, but the genetic material is being taken from the S strain, heat killed S strain to R strain, which is making the R strain virulent over here. Okay. So, here there is no contact between donor and recipient. This raises out the concept that is called as competence, which has the ability to pick the DNA from the solution. Okay. So, till here it is clear, right ma? Lastly, about conjugation and transduction also we will discuss. Conjugation, which is given by Liderberg and Tatum. Here this is the primitive type of DNA transfer. So, here if you see in the plasmid DNA, if it has the F strain, then it is, the bacteria is called as F strain bacteria. F strain bacteria, F strain, the F factor is transmitted from the F plus strain to the F minus strain. Okay. So, in the products or the daughter cells, both daughter cells will be F plus and F plus strains. Next, HFR. If F factor is present in the genomic DNA, if F factor is present in the genomic DNA, then it is called as HFR strain. When that F factor is transmitted to the F minus, then the products we get is HFR and F plus strain. Clear, right, about this two, F plus and F minus, HFR strain and F minus. Clear right now? So, here conjugation is possible only because of that sex pili, true pili, which is also called as conjugation tube in between F plus strain and F minus strain, or HFR strain and F minus strain. What are the products that we are getting is important, and you have to clearly know the difference between F plus strain and HFR. F plus means what beta? F factor is present inside the plasmid DNA. HFR means F factor is present inside the genomic DNA. Till here it is fine, right? Next, transduction, which is proposed by Zinder and Liedenberg. Okay, Zinder and Liedenberg, which is done on Salmonella typhimurium. If the genetic material is transformed through a bacteriophage, okay, genetic material injected into the bacteria or injected into the host through what? Genetic material injected to the host through the bacteriophage, then it is called as transduction. Best example is Salmonella typhimurium. So, here if you see useful bacteria and harmful bacteria. Okay. Useful bacteria, first for making curd from milk, lactobacillus is the example, production of antibiotics, Bacillus subtilis, nitrogen fixation, Rhizobium and Frankia. Here if you see harmful bacteria, chlorella causes Vibrio cholerae, typhoid Salmonella typhi, tetanus Clostridium tetani, citrus canker caused by Xanthomonas citri. So, all these examples are very much important. So, with this we have completed the Monera. I have already also told you from where the questionnaire actually arises. Each and every point which is keenly important for the NEET I have dealt. Each and every point you have to note. So, in the next class we will be starting with the next kingdom, that is Protista. So, that is all for today and thank you.